computational drug discovery pipelines. Adria works for Acellera, which is a company, I believe, largely based in Spain, but I think from their website, they also have a branch in the US. So Dr. Adria Perez graduated in biochemistry from the Autonomous University of Barcelona. And then he did a master's degree in bioinformatics for health sciences at Pompeu Fabra University. And after that, in 2017, he joined the computational science laboratory also at Pompeu Fabra University, led by Professor Gianni De Fabritis. And that's where Adria obtained his PhD in biomedicine. During his PhD, Adria's research focused on improving methods for molecular dynamics simulations through new machine learning algorithms and their application to protein dynamics. And as part of his doctorate, he also developed something called Adaptive Bandit, such a good name, and which is a new sampling algorithm based on reinforcement learning. And this was successfully applied in the study of disordered proteins. And Adria also worked on the development of TorchMD, which is a platform for creating and applying neural network-based potentials. And this has been particularly applied in coarse-grained potentials. And now Adria works at Acellera as a computational scientist, where he works both on the development of MD-based method and their application in drug discovery. So, Adria, the floor is yours. Thanks again for accepting to do this talk. We're very grateful for that. You have all the time you need, hopefully.
Okay, thank you. Thanks for the introduction and for letting me make this presentation here. All right, let me share my screen and start. No, not this one. Ah, yeah, this one. Great. All right. So welcome everyone. Today I'm going to speak about, in behalf of my company, Acellera Therapeutics, and I'm going to talk about the main applications that we use MD simulations for, particularly in drug discovery pipelines. So first of all, a bit of an introduction to the company. So we're talking about Acellera Therapeutics, which is a newly formed company. It's a US-based company, but the offices are located in Barcelona. And Acellera Therapeutics comes from previous company Acellera, which has extensive experience with MD simulations and computational drug discovery. And the main objective of Acellera Therapeutics is to make drug discovery computable, and so to go from what currently is drug discovery to drug engineering, so to automate the whole drug discovery process and industrialize it and scaling up with the thanks of computation. And focusing now on the topic of these talks, right, which is MD simulations. Acellera has, from its beginning, it has been related with MD since the company started with AceMD, which was the first molecular dynamics code that operated on GPUs in 2009. And this software has changed now through the years, and the current version, which is AceMD3, was co-developed with OpenMM, and as a technical side, supports all the common force fields. We're also started to supporting machine learning potentials, which we'll talk about later in the presentation. And currently now ASMD, it's not just a simulations engine, but a whole platform which allows you to not only perform simulations, but also to prepare them, build them, parameterize ligands, and also later to analyze your trajectories. Just as an introduction, and specifically showing the Python frameworks that are behind, that are within ASMD platform, HTMD and Molecule Kit. These codes are available in GitHub for free for academic licenses. And this ecosystem of Python libraries are meant to manipulate all types of files related with structural biology, like PDB files, small molecules, and mostly to prepare and build your simulations, and then later process the trajectories, analyze them, and do all sorts of analysis, like Markov state model analysis, and also scheduling your simulations. We'll see about this, since most of the applications that you'll see in today's presentation have been mainly built with these tools in ASMD platform. Just a bit of advertisement here, but now just as a summary of what we'll see here today. We'll see these three main applications of MD simulations for drug discovery specifically. So we'll start about talking about mixed solvent MD simulations. Then we'll go into high throughput MD simulations. So performing MD simulations for slower timescale events. We'll see more details later. And then we'll end up talking about binding free energy estimation methods. So let's start right away. Let's start talking about mixed solvent MD simulations. For those that don't know about mixed solvent MD simulations, it's where basically these type of simulations consist of trajectories of your target protein and several small molecules inside a simulation box. These small molecules tend to be like molecular probes or very small fragments, right? Such as a benzene ring, ethanol molecules. That's why it's called mixed solvent MD, because sometimes they're even solvent-like molecules. And this type of trajectories, so you have all this amount of molecules in your system, and so you can speed up the interactions between these small molecules and the protein, and also have much more interactions since you have much more molecules. And then you can see all sorts of characteristics for your system by the interaction of these small molecules. And the main applications that we use this for are for drugability assessment, mainly to find pockets and characterize pockets of different protein targets, and also to do fragment screening or fragment experiments, similarly to the fragment soaking crystallization methods that exist in drug discovery, but this time performed with MD simulations. So just staying in silico, going fully computational. And we'll talk about this now. First of all, the first application of drugability and exploring cryptic and allosteric binding pockets. And this is relevant for drug discovery in several ways. I mean, it's a tool to try to find new opportunities for existing targets, particularly to trying to find allosteric pockets. So in this way, you can either find ways to drug targets that are hard to drug, because the crystal structure that is available on PDB just has a main pocket which is known to be hard to drug. So you might try to find alternative strategies, alternative regions of the protein to target. It can also be interesting to overcome existing IP and patented space in a target, or even it's quite interesting, like for example, in case of GPCRs, to try to avoid, or on kinases as well, to try to avoid selectivity issues, and being very specific into a particular protein. And yeah, essentially we use this method to not only to find pockets, but also to characterize them with different pharmacophoric characterizations. So we typically do this type of simulations with different types of probes, like benzene or ethanol, or charged probes, so that we can not only find pockets that are not shown directly in the crystal structure. No, so for example, like with dynamics, and these probes, it helps to open up with a bit of induced fit. Sometimes open up pockets that are not shown on the starting structure, but also by analyzing the time and how the probes fit in this pocket, then we can later extract some pharmacophoric features out of these MD trajectories. And that is an application that we use very regularly, and that it works quite well. And then we can use these pharmacophoric features later in virtual screening and for scoring, which are interesting because they're derived from these MD simulations. And now continuing to the other type of application, which is these fragment simulations. This is an application that we can use mixed-solve MD for binding mode prediction. And in this way, what we do is that we prepare these mixed-solve MD systems, but with slightly larger molecules than these common probes that are used, so with a bit more complexity, and usually resembling to some scaffold of interest that we're working on our target, on our drug discovery project. And this can be helpful in order to identify a binding mode that we are not super confident of it, or we don't even know where it binds. This might start to hint. And the idea is that, well, we use fragments because they're small, so therefore the kinetics of binding are much faster. So the timescales are much faster. So then we're able to sample them with just trajectories of hundreds of nanoseconds, instead of having to invest hundreds of microseconds of simulation time, which this is quite a lot of computational time. So if we chunk out our ligands into small scaffolds, we can try to make these binding mode predictions, binding mode refinements of these smaller fragments. And here, just to showcase a practical example, for example, here we can take this compound here that binds into this protein. So we might be interested in the core scaffold here that we're developing, right? So we extract this fragment, and then we build these type of systems, right, with several copies of this small molecule. It's important that if they're very hydrophobic cores, to ensure that there's no stacking of them. And for example, on this case, with 5 replicas of 120 nanoseconds was enough. And then with the simulations, then we assess which ones are the most stable binding events. So all those poses and hotspots that remain there for a longer time in our simulations. And then this serves as a validation example, right? Because we knew this example was a crystal structure, we overlapped the pose we obtained with this type of simulations, and overlapping well with the native pose of the ligand from the crystal structure. And just to talk a bit about the types of probes of fragments that we use, it's important to note that we mostly use these type of probes. So the ones showed here, these are very small, and they're mainly used either for pocket discovery or pocket characterization, or for obtaining like pharmacophoric features for later scoring, because theirs are very small, and the binding kinetics here are always fast, and they bind everywhere. It's not really useful for later development of ligands, right? For that, we do use small fragments. So we try to make this type of small fragments for more binding, more prediction, or like simulating some scaffold that we might want later to develop. So these type of fragments usually are a bit larger. They have some much more moieties and some, at least up to one rotatable bond. But then, like really, what the word fragments is more commonly used in the context of drug discovery. The idea of a fragment is usually a bit larger, especially for experimental methods. But here, this method tends to have more limitations. Once the ligand becomes much larger and it has like more than one rotatable bond, then this method fails a bit, since the binding kinetics then are a bit slowed down, and the binding events that you'll get with your fragments are just very superficial and might not then correlate well with the exact binding modes, even that the fragment would have in a crystal structure. We mostly use this type of probes or smaller fragments that are quite rigid and very flexible. And we have several studies that showcase this type of virtual screening. I mean, it's a virtual screening that it's quite limited in the number of molecules that you test. It's not comparable to the undocking, but we have these two studies where we used a library of several fragments, here even in the hundreds of fragments, and from these we were able to develop some compounds that had some affinity, the ones that we show here in study. Also internally, we have used this successfully to obtain also some number of hits of a smaller size. But in general, we've demonstrated that some of these fragments are able to, and this method is able to recover quite well the binding mode of these fragments experimentally. And then here I also included this study here. It's not exactly virtual screening, or it's not using this method, this ligand binding by MD on the D3R receptor. So now I want to show you this video here. It's also a very nice video. I always want to show these on presentations. It's a bit old. It's from 2018, but it's super nice to see. And in this case, it's not a mixed salt MD, since we only have one copy of the ligand. But what we can see here, it's a much slower binding event, right? This is this type of events are harder to obtain with just simple trajectories, right? Since this accounts for several microseconds, and this type of time scales and computational time is not easily achievable, and unless you have specialized hardware like Anton supercomputer, this is hard to obtain with just single trajectories. And that's why this is my way to introduce the second application of MD simulations, which is what I call generically high throughput MD simulations. And with this, basically, I just referring to the ability of parallelizing your MD simulations. So yeah, as I explained, reaching longer timescales with single long trajectories takes too much time. Even with the most recent SMD update in the latest GPUs, reaching some simulation times of hundreds of microseconds, which is not that much at the time we are. You would need 4 months on an example system of factor 9, which is around 9K atoms, I believe. Well, considering this simulation speed, which is nanoseconds a day, you would take 4 months to just sample 100 microseconds. So this is not a way to go. And instead, what we can do is parallelize this sampling, having multiple short trajectories instead of having a single long one, or fewer replicas but very long. There's some methods that allow us to do that, because obviously short trajectories wouldn't be able to reach these slower timescales because they're all very short. But leveraging both Markov state model analysis and the possibility also, because we're doing parallel simulations, to optimize our sampling, deciding where we start the new rounds of simulations. This will allow us to reach more slower timescales, such as the one we've seen before. So yeah, now I want to introduce a bit Markov state model analysis for all those that might not know what this is about, which I consider that are very important for utilizing molecular dynamic simulations to their maximum exponent. So we have this example here. Now let's say that we have this set of parallel trajectories, right? Let's say we have these 3 trajectories with different frames at different lag times, right? And so first of all, what we want to do with the Markov state model analysis is to obtain a dynamic model of what is going on here, right? Because we have several parallel trajectories, it's very hard to just visualize them and make sense of all of them and trying to connect now, right? We can see here, this is some folding simulations, right? And here we have a trajectory that starts completely unfolded, and with different parallel trajectories, we sample the whole folding event. But this is very hard to connect without any help. So that's why we want to build this Markov state model, to obtain a complete dynamic model of what is going on in our simulations, to understand what's going on in our simulations, extracting the most relevant information out here. So first of all, what we're going to do is we're going to start by clustering our trajectories. This clustering, I mean, there's different ways of doing that, and it can be done using different metrics or factorizations like atom distances, dihedrals, contacts. Depends mostly on the application. But once we have our clustering here, what we can do is we can count the transitions we're having in our trajectories from one cluster to the other and obtain this matrix of transition counts, which then we can estimate into a transition probability matrix, which is essentially what the Markov state models are, this transition probability matrix. And then from this matrix, we can extract a lot of different properties, and we can obtain, we can model in different coarse-grained levels what's going on in our simulation, so we can understand. And for example, from these folding trajectories, we can recover the whole folding pathway, setting different metastable states, and also the different probabilities between them, and the flux and transitions between them. You can also obtain different estimations of binding free energy, binding affinity predictions, kinetic properties, and so on. And then also going back to what we said before on the opportunity to optimize sampling. Here are some, this is just some example to showcase the importance of this. So let's say that we start our simulation with a single trajectory starting from here, from the unfolded state, right? So yeah, our simulation then extends, these are our like starting points, then extends to this, and some trajectories, they reach this intermediate state. So with naive sampling, what we would do is we would just restart new simulations from, again, the same starting points. No, this is the whole explored space. So the second batch of simulations, we just start again from the same point. But yeah, doing this doesn't help in exploring more space. Like this depends a lot on your luck, and actually you're narrowing down your luck by not sampling longer trajectories in this way. So really to optimize well this high throughput MD simulations and this parallelization, it's important that you perform some decision making here and you try to optimize your sampling. So for example, there's what we call adaptive sampling, right? What we do is once we have our first batch of simulations and we have this explored space, then we make a decision. We say, okay, let's start our trajectories from more interesting places, right? So if then we start new batch of trajectories from this point, then it's easier for us to find more states, like in this case, the folded state. Yeah, here also I have this GIF showcasing what goes on in this adaptive sampling. So yeah, adaptive sampling works as by performing several rounds of independent parallel simulations, right? And at each round, at each epoch, what you do is you stop and you assess. Well, yeah, these are the simulations going on. So you stop, you analyze the simulation data that you have, and then you decide, what you can perform, like for example, some clustering and say, oh, okay, so from this state we have this is the state that we have the least exploration. So then we're gonna continue now sampling this this state because we have less information here. But really, the important thing here is that you decide and you try to point and direct the sampling into more interesting regions. The decision making can be any, really, and it can be either explorative, right? Trying to optimize sampling, trying to explore all the least sampled areas, trying to find new states, but also they can be exploitative decisions, right? More greedy approaches that try to focus on some scoring or some gradients. Like for example, we know what we're simulating a protein, and we know that there's a conformation, but we want to sample or simulate the whole event of going there, right? So we put as a goal RMSD to that structure, and this will help in deciding, okay, so from all the states that we have, which ones are the most close to that transition state that we want to go, and then select the states that are going to favor that we reach that desired state. And not only that, like you can have explorative or exploitative decisions, but it also the balance of it that it's important, balancing correctly exploration and exploitation. And here at Acellera we have several algorithms that deal with this, but I don't want to go much more into this. And so after having explained what adaptive sampling and MSM analysis are, these are the base of what we call our in silico assays. So we have in silico conformational assays and also binding assays. And essentially we refer to these as more extensive MD studies, where we take, for example, in silico conformational assays, we take one protein and we perform extensive simulations in the order of tens or hundreds of microseconds to try to sample as much the conformational dynamics and the conformational space of this protein. And so, as I said, we use adaptive sampling and Markov state models. And after these simulations, we are able to obtain a dynamic model of our target. And this can be very important during drug discovery campaigns. Understanding the dynamics of your protein can be critical, depending on the protein, since the protein function depends on its dynamics, and this dynamic information is something that we do not have when we only look at the experimental structure. So with this method of performing several rounds of parallel simulations, we're able to obtain complete characterizations of the dynamics of one protein. And this is useful in drug discovery for several ways. Like we can compare different metastable states of our protein. Perhaps there's we can compare active and inactive states. We can also compare different systems through different mutations, see if a mutation, how it affects the dynamics of our structure, which can be, it can, in some cases with MD, it might be the only way that you can completely assess, besides experimental, obviously, but it can be the only computational way that you have to assess the effect of a mutation in your system. And in the same way that we have our conformational assays, we also have in silico binding assays. So in the similar fashion of extensive simulations of the order of hundreds of microseconds, here instead our objective is to simulate a whole binding event between a protein and a small molecule. And by simulating the whole binding event, then with Markov state model analysis, you don't only obtain like a full model of the binding event and the different metastable states, but you can predict different properties such as the binding affinity and different kinetic properties such as K-on and K-offs. K-offs are a bit more harder to obtain since usually for good ligands, these are quite high and then therefore like very slow timescales, and therefore they're very hard to sample. But for binding affinities and K-ons, this is perfectly doable. And yeah, now I'm going to show you some case studies that we have using this type of high throughput MD simulation. So first one, it's a study that we did in collaboration with UCB, which is, yeah, we applied ISCA, which is the in silico conformational assay, to GPCR receptor A2A, and basically UCB came to us because they wanted to sample the I think the activation pathway of a GPCR, no, going from an inactive to an active state. And yeah, we used adaptive sampling and some goal-oriented scoring function to guide the simulations unbiased to a specific active conformation, to the activation pathway, exactly. And then we were able to obtain, yeah, different metastable states out of the MSM analysis, and you are also able to look into details into this activation pathway. And for example, in this example, it was important to understand the influence of cholesterol in here. And yeah, all this information can provide you with very important information for your drug discovery campaigns. Yeah. And then for case study 2, this is the one that I showed before with the video. So it's the same example of the D3R receptor and the ligand. So this was a collaboration done with Pfizer some years ago, where they had this case that they have this novel antagonist for D3R receptor, and they have these antagonists worked, but they were not seeing correlations with the binding mode that they had, that they obtained through docking, and then the experiments that they were producing and the compounds they were producing based on that binding mode prediction from docking. And this is, we understood later, because the binding mode from docking was completely wrong, because with the simulations and these extensive simulations, we were able to find the correct binding mode of this ligand, which was in a buried cryptic pocket that wasn't previously known for this target. So in this example, we have this example where that highlights the importance of dynamics here in MD and what it can bring to a drug discovery campaign. And because this binding mode would have not been able to obtain this without the use of MD simulations. And I mean, not only you get this binding mode prediction, but you also, again, are able to characterize the whole binding event. So you can identify the different metastable states, which can help you identify what are the important groups and moieties in your compound for then further development. So yeah, with this type of simulations, you can obtain very fruitful description of the dynamics of your system. And then going next to this case, the third case study. Here I wanted to show an example that showcased intrinsically disordered proteins, which might not be as related with drug discovery yet, but definitely if you're dealing with intrinsically disordered proteins or IDPs, you're probably going to need MD simulations if you want to deal with them computationally, obviously. Because here in the case of IDPs, a single structure, single crystal structure, or even NMR structures, which only have a few frames, might not be enough to characterize completely the structure and dynamics of this type of proteins, which don't have a specific conformation. And these dynamics and this fluctuating structure, it's key to their function, right? So the only computational way to understand these IDPs is through MD simulations. And here, showcasing what this can do, I'm showing here an example on this study here where we simulated the IDP CMIP, which is the small one here, with the protein KIX. And we simulated the whole binding event of these two proteins, the IDP and the protein. And what we also were able to see with the simulations, not only the binding, but at the same time the folding of the IDP. So the IDP has an unstructured state, or and in solution, no, it sometimes has this small helical part here, but yeah, this fluctuates a lot. But then what we can see through this binding is how the IDP first does the first contacts and then starts folding into a complete helix to the final bound state. And with this type of high throughput simulations with adaptive sampling and with Markov state model analysis, you're able to obtain these, get this complete dynamic description. And not only that, but you can go even beyond and you can look more in detail into these macro states, metastable states. So yeah, showing these macro states here, 13 and 11, we wanted to know, okay, how does this process go? Is this goes through conformational selection, or is this an induced fit, which is a common question, particularly with IDPs, on how they work. And what we were able to see analyzing the flux between so we discretized, yeah, we separated these macrostate 13 and 11, and we separated them, clustering them through their secondary structure. So the part that had helical parts or completely unfolded, and we saw that most of the flux in the first state, so the first where it starts, it does the first contacts, is prioritized by conformational selection. So you have helical conformations in the end terminal of CMIP. Then these interactions are much favored to then go through the whole binding unfolding event. And yeah, this I mean, overall, the mechanism is induced fit, but there's this initial conformational selection. And all this information, you're able to obtain it thanks to this type of analysis, which, yeah, it's very powerful. And yeah, summarizing also this, to summarize, we've done different studies on IDPs, and not only the example we showcase here, but also with MD, we were able to, you can also just characterize an IDP in solution, see the different states that it can, that the structural and secondary structure that it can form. But also we also have this study where we simulated the interaction between a small molecule and an IDP, which might be of interest for more experimental developments in drug discovery. But yeah, that's all for high throughput simulations. Let me just drink some water. Yeah, before going into binding free energy estimation methods, which is one of the main applications of MD simulations in the drug discovery industry. I'm sure that most of you know about them. We have several different studies and several different applications that are that have ways to that perform binding affinity predictions, from MD-based ones, here the MSM ones, just as the one that we showcase with the D3R receptor, or other similar ones. But also here we have some studies that we do that with machine learning methods. This is not exactly simulations, but still related. But mostly what we're going to talk about now in this section is yeah, relative binding binding free energy estimations, mostly focused on relative binding free energy estimations. And here we have some papers trying to validate this specific method, which is ATM, or alchemical transfer method. So this method wasn't developed by us, but developed by Emilio Galicchio's lab, and but we did work with them to help them validate this method, which I'm going to show now the results. But first, a small introduction on what the alchemical transfer method is. So the interesting thing about this free energy prediction method is that, differently from other common ones like FEP, and here we have a single simulation box. And in this single simulation box, we put the protein and 2 ligands, one in the binding pocket and one in solution. So in here, instead of having 4 different systems, one with the ligand in solution, one with the ligand bound, and the same for the other 2, you have only a single system, and this simplifies a lot the execution of the simulations. And essentially what this method does is that, it interchanges the ligand between them. So you perform these different lambda annealings, where basically you exchange the ligand between them. You have this this state at the middle, which is an intermediate state where the both ligands are present, both in solution and the ligand, and with replica exchange, then you perform the simulations, and then you're able to obtain the free energy estimates. And thus we worked with this code to validate this method. So first of all, we compared. We used the common benchmarks such as the Schrödinger and Merck datasets, and we compared them. I forgot to mention one very important thing is because we only have a single simulation box, right? This means that we can use any type of force field for these simulations, which, in comparison to methods like FEP or other methods which require specific edits of the force field or custom force fields for each system. Here it's much easier because you have a single simulation box, and then you can just use Amber or any force field. So it's important here because what we did is comparing different force fields, mainly GAF2 and OpenFF, and we also compared it to other methods such as FEP and PMX. And from what we can see from this evaluation is that from the force fields, the one that works best is GAF2, which provides some quite good results. It's not as good as FEP. In terms of correlation, it works quite well. It's not as good as FEP, but it's much simpler to use. And it does have a tendency to have larger mean absolute errors, and this, I think, it comes with the method. In terms of correlation, it works quite well. And specifically, we saw this for GAF2, for the OpenFF force field. At the moment that we tested this, was not working as well. I think that they're working on this. So, because we're able to use any type of force field, this gives us an opportunity to use this new advancements, which are neural network potentials. And the idea here is that we would use neural network potentials to parameterize the small molecules, since they have been training with QM data and they are much more precise at parameterizing these ligands. And the ligand force fields is like one of the main contributions to the accuracy of these simulations. So we also performed an evaluation using this method. But how does this method work? Well, first of all, just wanted to introduce a bit neural network potentials for those of you that might not know what this is. So essentially, what we do here with neural network potentials is that we use deep learning models to approximate forces and energies out of coordinates. So in the same way that common force fields and force fields use, that from the coordinates of your system, you compute the forces and the energy. You do the same, but using a neural network. So you train it with different atomic coordinates and the corresponding forces and energies. And then you're able to obtain this neural network potential, which its strength is that it's able to learn much better the many-body interactions, and it's able to do a much better job at doing this inference than common force fields. So the idea here is that for MD simulations, what you do is you train a neural network potential on QM data, which is much more accurate than molecular mechanics derived force fields. And then you train a network which is much faster than QM methods. And thus you can reach the accuracy of QM, but with speeds more similar to normal ND simulations with molecular mechanics force fields. That's the overall summary. And how we can perform simulations is because we worked to enable in ASMD the ability to perform a mixed NNP and molecular mechanics simulations. So similarly to what is done with QMMM, basically you use the network potential to parameterize and obtain the forces and energies of a small molecule, and for the rest of the system you just use standard molecular mechanics like AMBER, for example. And therefore we use this method but applied to alchemical transfer method and relative binding free energy estimation. And we evaluated this method compared to the previous method just using GAF, so just using molecular mechanics, and also to FEP. And what we can see here is by using these neural network potentials and these NNP-MM simulations is that we can overall obtain a very good performance of ATM. In this example here, we used Ani2X as the neural network potential. And yeah, what we can see is by using Ani2X, it definitely improves the performance of GAF2, and it's more or less on par or even better in some cases than FEP. We have this example where it's performing much worse. But I mean, the idea is that these are very promising results because the interesting thing about neural network potentials is that they can be improved with better training and much more training data. So there's a lot of room for improvement on neural network potentials, and we are already obtaining these type of performances. And also, most of the mean absolute error is lower than 1 kcal, which is great for the type of calculations that we do here and for predicting binding free energies. The main drawback is, though, the speed of the simulations, since network inference and using neural networks is still not as fast as using just plain MD simulations engines, and it's a trade-off in speed accuracy, but it's not impossible as if we were using QM calculations. So yeah, and this is very promising, but Ani2X is a quite limited neural network potential. And for example, one of the limitations is that it cannot deal with charged molecules. So currently, it's not viable to be applied in drug discovery campaigns. So that's why in Acellera we're working on our own neural network potential, and we're building our own data set. Well, the neural potential is called AceForce, and we're building our own training data set with different chemical structures from PubChem. We're performing QM calculations at this level of theory with a diverse set. This is just some conditions that we're using to filter different compounds. Now we're focusing on smaller molecules, and we're building up our neural network potential step by step. But yeah, we have already performed some tests with this first version of the data set. And we already done some evaluations on relative binding free energy in the same way that we did with Ani2X. And what we can see here is that we're obtaining similar performance to Ani2X. We're consistently better than GAF2. But the benefit here of using AceForce is that we were able to deal with some systems that had charged molecules that we were not able to deal before with Ani2X. So if you can see, a lot of these systems here that don't have a value for Ani2X is because Ani2X cannot deal with them because it has not been trained with charged molecules. It doesn't predict them well. So yeah, with AceForce, we're keeping more or less the same performance with this first version of AceForce, but already fixing some of the issues with other neural network potentials. And yeah, also one interesting metric to look at is the fraction of top scoring compounds. Since one of the main applications, well, the main users of binding free energy estimations in industry, they mostly care about ranking of compounds. not so much about the specific accuracy of the method. No, so if your method, even though like the free energy are not, there's an error always in the prediction, if you're consistently able to detect the top compounds and differentiate the top compounds and the worst compounds of an entire set of molecules, that's what it's most interesting for the industrial application of these type of free energy predictions. And here we looked at these statistics for this evaluation. Yeah, this is some preliminary evaluation. We're gonna do a more thorough evaluation, and we're preparing a publication here. But just looking at the results we have so far, we can already see that ACE-Force consistently does well in detecting the top 5% of compounds and top 13% of compounds, especially compared to FEP, and definitely much better than GAF2 in most of the systems. So yeah, that's some very nice results we're getting here. And so yeah, overall, summarizing this, from Acellera, we provide this type of solution for relative binding free energy calculations, which is found within the Quantum Bind platform, which I'm going to talk about now in a few slides. But yeah, just to summarize, with relative binding free energy calculations, this is mainly used in lead optimization stages to compare similar compounds. So similarly structured compounds. You can also perform scaffold hopping, since ATM, the alchemical transfer method, allows you to perform scaffold hopping comparisons. And we provide the way to test your congeneric series, either with molecular mechanics force fields or with NMPs. So we provide you with this trade-off between speed and accuracy, right? So with GAF2 molecular mechanics, you're able to screen a much larger library of compounds, but with NMP you can be much more accurate. So we tend to do a combination of both, and we use NMP for those predictions that we want to be more secure of. But yeah, depending on the system, the simulations with NMP, it can ramp up to a lot of hours and slow down a lot the prediction of this. And also, we've been working now recently with a method to do absolute binding free energy calculations. So this, you don't do a comparison between two ligands, you just do a single ligand directly. It's currently under internal validation and development, but the first tests have yielded already some interesting results. Here's an example test with an internal target that we have, which is RIPK2. And for a list of molecules that we send out to that we tested, we used the ABFE method to predict the delta G's, and we yield a threshold here to define what we detect as positive and negative. And what we've seen that we like here is that from the ones that we labeled as negative, so the ones that we labeled not good enough, we only find one case of false negative. So it seems that we were able, from the whole list we have here, the method was able to find most of the hits, and mostly the ones that yield a higher potency later in experimental results. We can see then that they're ranked on the top. But yeah, this is still under internal validation, and we'll release more results as they come by. Still work in progress. But yeah, just to end up a bit talking about Quantum Bind platform. Quantum Bind platform is not only about binding affinity calculations, but it's a whole discovery platform where it combines state-of-the-art generative chemistry to generate libraries of molecules optimized to the conditions and your drug design objectives. And it's all combined into an active learning cycle that I'm going to show you here, where you continuously select compounds from a large library that you might have generated or that you have from your property. And you continuously select compounds here to perform binding affinity calculations. It can be relative or it can be absolute. And since these are expensive, you just set some of them, and then you use these results to train a machine learning model that you will use to rank your library, which is very fast, right? And then select the next compounds to test, so to prioritize in this large library. So the idea is that with this active learning cycle, the first rounds are used to just generate some calculations to train the ML models, so that this is it becomes much more precise and much more specific to your target. And then once this gets better and better, you're better at screening your large library of compounds, prioritizing compounds to validate them with the methods, and also in between rounds, you just perform experimental testing of the most interesting compounds to make sure that the estimations that you're doing, either with simulations or with machine learning models, are being correct. And after this, this is all my presentation and what we had to present about our applications on MD simulations. So yeah, thank you all for your attention. And if you have questions, I'll be gladly asking.